slide or because you have a
Hello students, a warm welcome to all of you. Today I am going to discuss about the history of immunology. Earlier we have already discussed regarding the development of the microbiology through the commendable work of some of the most notable scientists in our world. So we know that the foundation of immunology, which is a field department of microbiology, was laid in the late 18th century in the year 1796 when the Edward Jenner developed the vaccine against the smallpox. Now, for 1 century, there was no much development in this field of vaccine or immunology before Robert Koch and Louis Pasteur made some important discoveries. Louis Pasteur, if I say, his work on chicken cholera, rabies, and anthrax vaccine laid the foundation of immunology as well as the Koch phenomenon, which was basically on the hypersensitivity reaction on tuberculosis against the tuberculosis, had made this field an interesting and a very important to treat infection. So as we see that in the till mid 18th and in the early 19th century, we have already discussed that so many bacteria were developed or founded who were responsible for different type of infections and diseases. On the similar pattern, the work was also going on in the field of immunology, means that how our body reacts against these foreign pathogens and how our defense system works. So in this chapter, we will discuss about the work of some notable scientists in this field and some of the important Nobel laureates who got Nobel Prize for their work. And also it was a landmark in the field to treat in the field of vaccine as well as for the treatment of some of the most important pathogens or infections. So we will see some of the work of the some important scientists whose work is regarded as one of the milestone in the field of immunology. So first scientist which I am going to discuss today is about George Henry Falconer Natal, year 1862 to 1937, his lifespan. He was an American-born British biologist. Natal in the year 1888 was the first to demonstrate that defibrinated blood possessed a considerable bactericidal power against anthrax bacilli. So basically what he did that he removed the clotting factors from the blood, which helps in the clotting of the blood, means that fibrin was removed, and he showed that when the defibrinated blood was used against the anthrax bacillus, it has a very bactericidal, means it was working against the bacteria or killing those bacteria. And also an interesting work was done that at this the power was destroyed, that the power to destroy the bacteria by the blood, defibrinated blood, was destroyed at 55 degrees centigrade, and they also disappeared with the time. So the work formed the basis of an essay that a contribution to the study of immunity, for which Natal received the triennial Wollaston Prize from Harvard University open to competitors of all nations. The essential facts disclosed by Natal were confirmed by many workers who showed further that the power varied with the bacteria employed. Next important work was done by Hans Ernst August Buchner, 1850 to 1902, a German bacteriologist, and his work on immunological studies span was from the year 1886 to 90, and he discovered a naturally occurring substance in the blood now known as complement. So Natal discovered that the blood was defermented blood was basically had some bactericidal effect which get destroyed at 55 heating at 55 degrees centigrade, whereas Hans Ernst Buchner discovered a substance that was later came to be known as the complement, and that is also capable of destroying the bacteria. Next important work he was on is methods of studying anaerobic bacteria. Buchner was one of the first to note that a substance in blood serum, alexin, could destroy bacteria. The substance, renamed complement by the immunologist Paul Ehrlich, is now known to be the part of the complement system, which consists of about 20 proteins that act together to eliminate infectious organisms from the body. Basically, these complement proteins acts in the form of a cascade in a chain of reactions, and any bacteria which is recognized by the host system as a foreign, so these complement gets activated, and step by step they work together and finally helps in the phagocytosis or the destruction of the bacteria, and it is a part of our blood. Next important work which was done by the scientist was Eli Mechnikov, year 1845 to 1916, Russian-born biologist working at the Bacteriological Institute Odessa, 1886 to 1887, and at the Pasteur Institute Paris from 1888 to 1916. Mechnikov contributed to many important discoveries about the immune response, host's immune response, how they act against any of the foreign substances. And his work, perhaps his most notable achievement, was his recognition that the phagocyte is the first line of defense against acute infection in most animals, including humans, whose phagocytes are one type of leukocytes or white blood cells. So first, we see that Buchner and et al. Showed that some proteins or some substances are present in the blood which have some bactericidal effect, whereas Mechnikov discovered a substance called as the phagocyte, which is also a part which he concluded that it is a type of white blood cells. And this work formed the basis of Mechnikov's cellular theory of immunity in 1892, a hypothesis that engendered much opposition, particularly from scientists who claim that only body fluids and soluble substances in the blood, which is basically known as antibodies, and not cells destroyed invading microorganism. That is the which constitute the part of the humoral theory of the immunity, who received with in the 1908 Nobel Prize for Physiology or Medicine for his discovery in animals of amoeba-like cells that engulf foreign bodies such as bacteria, a phenomenon known as phagocytosis and a fundamental part of the immune system. Earlier it was thought, or the concept was that only the soluble part of the blood is only responsible for the immunity, basically, which is the part of the antibodies. But Mechnikov discovered that even blood cells also are involved in the defense system of the body, and especially by phagocytosing the foreign cells or the pathogens which constituted the cellular. Basically, we divide immunity into two types, that is the cellular part and humoral immunity. Humoral immunity, basically, it consider, or it is based on the antibodies or the soluble liquid part of the blood which is involved in the immunity, whereas the cellular immunity consider or emphasizes on the work of the cells which are mostly involved in the immune mechanism. Next important work was done by Emil Adolf von Behring and Tsubasa Buro Kitasato. Emil von Behring and Tsubasa Buro Kitasato collaborated to discover that serum from animals infected with tetanus could provide immunity to other animals. So it was also a type of which we say that a type of passive immunity, and first work was done by von Behring and Shibasa Kitasato, who discovered that when any animal who was infected with tetanus bacteria can, and when their serum was extracted, they can provide immunity to other animals. And this research, published in 1890, led to the development of serum therapy and the concept of antitoxin. They formed the base of the serum therapy which are still in use now, and also the concept of antitoxins. Von Behring went on to win the first Nobel Prize for his work on diphtheria serum therapy. Basically, diphtheria causes infections by secretion of some toxins, and they can be neutralized by administering some antitoxins against them. So it was the first work which was done by on this field was by Emil von Behring, and Kitasato is recognized as a pioneer of Japanese medicine and co-discoverer of the antitoxin. So along with Shibasaburo Kitasato, which was a Japanese scientist, they together developed the concept of antitoxins which provide immunity against the toxins secreted by the bacteria. So what was the serum therapy, and how they discovered these antitoxins, and what was the pathway for the discovery? So we'll see a brief look on this part. So what happened that in 1890, Waring and Kitasato published their findings that serum from an animal with tetanus could neutralize the toxin, a discovery that laid the foundation for serum therapy. Kitasato had previously succeeded in the pure cultivation of the tetanus bacillus and in developing the antitoxin for it, which was the basis for their joint paper. So they worked together and they had a collaboration among them against the work of the how to neutralize the toxins, and uh, they together laid the foundation for the serum therapy. And this work established a method for passive immunization, which was a significant breakthrough for treating infectious disease. Even today, we see that, especially in the case of tetanus and diphtheria, we give anti-tetanus serum, ATS, or anti-diphtheritic serum. So for their treatment, basically, they are antitoxins, or we can say in other terms that antibodies prepared in some other individuals or animals, and further they give immunity against any type of infections, recent infections, and helps in providing the short-term immunity in the patient. Now, what was the individual contributions and legacy of Emil von Behring? So he applied the principles of their joint research to develop a successful diphtheria antitoxins, which led to him winning the first Nobel Prize in Physiology or Medicine in 1901. So Emil von Behring was the first person who developed the anti-diphtheritic serum, or we can say that diphtheria antitoxins. And for his work on diphtheria antitoxin led to winning a Nobel Prize in 1901 in physiology, or we can say medicine. He later developed a toxin antitoxin mixture to induce permanent immunity, and his work is the basis for modern immunization practices. The next important scientist work on antitoxin was Shibasaburo Kitasato, and is celebrated as a pioneer of Japanese medicine for his role in developing serum therapy for tetanus and diphtheria. So his work was on also concentrated on tetanus and diphtheria, and he established the Institute for Infectious Diseases in Japan and several as the first president of the Japan Medical Association. Kitasato's research philosophy, which emphasized clinical applicability, continues to inspire many Japanese medical researchers today also. So in Japan, he's one of the pioneer of developing the medical sciences, and still his work is recognized as one of the some of the most important works in the field of microbiology and immunology. Next, the work which we can say the important work was Richard Pfeiffer, a German physician and bacteriologist. Here you can see Pfeiffer is working with Robert Koch, and he is credited with the discovery of endotoxins. So we will see what is the endotoxin. So earlier, Kita Sato and Emil von Behring, who worked on toxins and antitoxin, Pfeiffer also worked on the same phenomenon, but here specialized himself in the field of endotoxins. So what are the endotoxins? So Pfeiffer is remembered for his many fundamental discoveries in immunology and bacteriology, particularly in the phenomenon of bacteriolysis. In 1894, he found that live cholera bacteria could be injected without ill effects into guinea pigs previously immunized against cholera, and that the blood plasma from these animals added to live cholera bacteria caused them to become motionless and to lyse. This could be inhibited by previously heating the blood plasma. He called the bacteriolysis, and it became known as Pfeiffer phenomenon or Isaiah-Pfeiffer phenomenon. Basically, his work was on the cholera vaccine, and he found that when the live cholera bacteria injected into a vaccinated pigs who were into guinea pigs who were previously immunized against the cholera, and that blood plasma from these animals added to live cholera bacteria caused them to become motionless and to lyse bacteria, and this could be inhibited by previously heating the blood plasma. He called this phenomenon as bacteriolysis, means that lysis of bacteria, but the bacteria though get lysed, but their toxins which were secreted were able to develop immunity again in the guinea pigs. So we can say that if he laid the foundation of the type of attenuation of the bacteria, means the bacteria though it loses its pathogenicity but retains its immunogenicity. So the he was the first to lay this foundation for the work on this field, and this phenomenon was known as the Pfeiffer phenomenon or Isaiah-Pfeiffer phenomenon. So working with Robert Koch in Berlin, he intellectually and experimentally conceived the concept of endotoxin as a heat-stable bacterial poison responsible for pathophysiological consequences of certain infectious diseases. Endotoxins and anti-endotoxin antibodies have since then fascinated researchers of many disciplines, particularly in the field of diagnosis, preventions, and therapy of severe gram-negative infections. If the students who are familiar with microbiology field, they would be knowing that there are two types of toxins, that is exotoxins and endotoxins, and endotoxins are mostly secreted by the gram negative bacteria, whereas exotoxins, which constitutes the part of earlier diphtheria and tetanus, are mostly secreted by the gram positive bacteria. So we can say that von Behring and Kitasato worked on exotoxins part, whereas Pfeiffer worked on endotoxins. So endotoxins are mostly secreted by the gram negative bacteria, such as cholera is an example of that. So we can say that it both work was commendable in developing the immunity in the individual against some of the deadly pathogens. So Pfeiffer was a pioneer in typhoid vaccination also. He discovered the specific bacteria dissolving immune bodies in cholera and typhus. The British pathologist Almroth Wright is generally credited with the initiation of typhoid vaccine in 1896. Earlier credit is given also to Almroth Wright, who also initiated the development of typhoid vaccine in 1896. His claims of priority were challenged as early as in 1907 in favor of Richard Pfeiffer. A review of the original literature in 1890s and the early 1900s revealed that several groups were working on typhoid vaccine at the same time. So, if the favor in the judgment was given in the favor of Richard Pfeiffer, but the work of Almroth Wright cannot be ignored because he first of all he discovered, and we see that during this period there was lot of work which was done in the field of the development of typhoid vaccinations through their own theories. So in 1897 Pfeiffer joined the German expedition under Robert Koch to India to investigate the plague. So he combined again with collaboration, or we can say that with he joined the expedition with Robert Koch to India to investigate the plague in 1897, and the following year he went to Italy with Koch to do research on malaria. So he was a disciple of Robert Koch, and not only on the endotoxins of cholera or typhoid, but his work is also on other bacteria also, including the plague and the protozoan parasite malaria. Next work, the scientist was Paul Ehrlich in the from the year life period 1854 to 1915, a pioneering German medical scientist in microbiology, immunology, and chemotherapy. His work on immunology was from year 1890 to 1906. Ehrlich joined Koch's Institute for Infectious Diseases in Berlin in 1891, where he collaborated with Emil von Behring on standardizing the production of an effective antitoxin serum for diphtheria. We have already discussed about von Behring and Kitasato, who laid the foundation of serum therapy, and on the their work for developing antitoxins against diphtheria and tetanus bacillus. So he worked along. He was the Paul Ehrlich also collaborated with Emil von Behring, and he standardized the production of an effective antitoxin serum for diphtheria. He formulated the side chain theory in the year 1897 to explain how cells bind to toxins and produce antibodies. So side chain theory was given by Paul Ehrlich in the year 1897 to explain how cells bind to toxins and they produce antitoxins or antibodies, a foundational concept in immunology for which he shared the Nobel Prize with in 1908 with Elie Machnikov. He also coined the term complement. Remember, the Alexin term earlier was given, and he was the person who coined the term by Machnikov, and he termed it as the complement in which helps in the elimination of the pathogen. So in 1899, to describe the heat labile component of serum involved in immune reactions, regarded in accordance with this conception, the antitoxins represent nothing more than side chains reproduced in excess during regeneration and therefore pushed off from the protoplasm, and so coming to exist in a free state. So what happens that when these antitoxins are nothing but they represent the side chains which are reproduced in excess during regeneration and therefore pushed off from the protoplasm, and so coming to exist in free state. When they are produced in excess, and they remain uh, they are pushed off from the protoplasmic state and remain in the free state. And when the pathogen attacks the body, they again develop a side chain development, and which prevent the any type of infection. And he got for this the Nobel Prize in the year 1908. So here you can see the side chain theory, that this theory was proposed by Ehrlich. So here you can see that according to this theory, immunocompetent cells have surface receptors. Here you can see there are different type of surface receptors which are present on the immunocompetent. Immunocompetent cells are those cells which are capable of producing immune cells. Now here have surface receptors that are capable of reacting with antigens and which have complementary side chains. Here you can see there are different type of complementary side chains. When antigens are introduced into the host, they combine with those cell receptors that have a complementary fit. So this is basically a lock and key mechanism where the antigens react with that particular complement, and when the antigens are reintroduced into the host, they combine with those cell receptors that have a complementary fit. Now this inactivates the receptors. There is an overproduction of the same type of antibodies, same type of receptors that circulate as antibodies. So this was the basic of side chain theory. Next important scientist whose work is recognized as one of the most important part of immunology field is Jules Baudet, year 1870 to 1961, a Belgian scientist. Jules Baudet joined the Pasteur Institute in April 1894, where he attended the Grand Cours de Microbie organized by Dr. Emily Rox. This gave him the opportunity to meet Elie Mechnikov, the father of cellular immunity. Very soon, Jules Baudet joined the Mechnikov's laboratory, in which he developed an independent line of research, which culminated in the seminal demonstration that killing of bacteria depends on interactions between antigens, antibodies, and complements. So he demonstrated that there is not only a single factor which is responsible for the killing of bacteria. It depends upon there is a mutual interaction between antigen, antibodies, and complement. Mechnikov rapidly recognized the importance of Baudet's contribution, which he already mentioned in his report to the International Congress of Budapest in 1894. It is during the same period that Jules Baudet deciphered essential mechanisms of agglutination and lysis of sensitized red blood cells. On October 28, 1920, Jules Baudet was awarded with the 1919 Nobel Prize for the discoveries relating to immunity, namely his work on the complement system. He deciphered the mechanism of the bacteriolytic activity of immune serum obtained in animals immunized with bacteria and the hemolysis capacity of anti-red blood cell immune sera. But he was also a distinguished bacteriologist who worked on bacteriophages and discovered the causative bacterium of whooping cough, named as Bordetella pertussis in his honor. So we can say that where the other scientists we have already discussed have their separate work on different parts. Some worked on the plasma proteins or blood proteins. Some worked on leukocytes or phagocytic cells. Some worked on antitoxins, toxins. But Jules Baudet was the first scientist who came to the conclusion that not only a single factor is responsible for the destruction of antigen or bacteria, but it is a combined action which involve bacteria, then proteins, blood proteins, as well as the cell also. Okay, complement parts, antibodies, and complement. Okay, so here he was the first scientist who widened the scope of immunology. He included or different parts of the immune system together. Even the work on the red blood cell immune serum means that how red blood cells interact with the anti-red blood cells. And his work was not only limited to the immunity or immunology. Other scientists also have their other works also, but his work is also considered very important in the discovery of important bacteria known as Bordetella pertussis, which itself is named in his honor, which is responsible for whooping cough. Next, scientist, we have already discussed about the typhoid vaccine. Work was initiated by him, and his name is Sir Almroth Edward Wright, 1861 to 1947, a British bacteriologist and immunologist. And he discovered his main work. His work is considered remarkable for the discovery of opsonin and the opsonic index unifying humoral and cellular immunity. In collaboration with Stewart Rankin Douglas, Wright discovered substances in the blood serum that coated bacteria and made them more susceptible to destruction by phagocytes, which is a white blood cells. He named these substances as opsonin. So he discovered that there are some substances which are known as opsonin. So they are responsible for coating the bacteria, which makes easier for the phagocytic cells to phagocytose them or to digest them. And he named these substances as opsonin. And this discovery helped to unify the previously separate theories of immunity championed by Elie Mechnikov, cellular immunity or phagocytosis, and Paul Ehrlich, humoral immunity or antibodies, showing a necessary cooperation between the body fluids and its cells. And what is the opsonic index? So he introduced the opsonic index, a measure of the body's capacity to prepare bacteria for phagocytosis. Basically, the measurement of the host cells capacity to uh, coat the bacteria, and the criteria for measurement of this capacity was named as opsonic index, which was used to monitor and control the dosage in his system of vaccine therapy using autogenous vaccines prepared from the patient's own bacteria to treat established infections. Next important work, which is done by Paul Potier in 1866, Paul Potier life period 1866 to 1962, and Charles R. Richet 1850 to 1935, a French scientist who important aspect of the immunity is also hypersensitivity reactions, or we can say anaphylaxis. So what was here, I have drawn a figure, or I have demonstrated a figure of a dog and a sea anemone, which we'll discuss later, which we'll discuss in the next slide, that what is the relationship and what was the experiment. So what happened is they also got the Nobel Prize for their work in 1913, awarded to Charles Richet, French physiologist, for study of anaphylaxis. So what did Richet and Potier found that injections from the nematocyst of Physalia induced a violent reaction in dogs, including the death of a fine big dog by the name Neptunus. The dog name was Neptunus. Remarkably, dogs experienced no apparent ill effects when they received only a single injection. So when the injection of this product or product of nematocyst Physalia was given to the dog, so there was no ill effects or no reaction shown by the dog. But as the second injection was given to them, these results harkened back to Richard's earlier observation of similar adverse symptoms with repeated injections of eel serum. So when they given those second time, so they found that there was a severe reaction in the dog of these product of Physalia, and finally it led to the death of the dog. And the investigators settled for a comparative study using sea anemones that were which were obtainable in large quantities of the rocky shores of Europe. They were successful in replicating their physiological result with an extract from the tentacles of actinia when injected into various experimental animals. Actinotoxins, as the extract was termed, which was extracted from the tentacles of actinia, So these actinotoxins produced dramatic results, especially in two dogs which died when a repeat injection was given 14 to 23 days after the first sensitizing dose. So the first dose here was called as the sensitizing dose. See here what the reaction takes place, that the when a person or any animal who is hypersensitive to some of the extracts of some product, plant product. So during the first dose, they are sensitized, means the immune system are prepared, and the first dose does not cause any ill effects. But as soon as the second dose is given, then a severe reaction, or we can say hypersensitive reaction, occurs in the body in the form of shock, just itching, dyspnea, vomiting, hypotension, stupor, and death in half an hour. In 1903, Rickett published his findings. While a foreign substance might induce a mild reaction upon first exposure, it could produce a severe hypersensitive symptoms and even death when reintroduced later. Instead of providing protection or prophylaxis, Rickett had discovered a new biological response he termed anaphylaxis or anti-protection. So here what happened, that instead of immune system, though during this period it was regarded as a had a protective functions, but here Rickett and Potier also showed the anti-protection activity of the immune system, which are even get very fatal and lead to the death also. Next important work was who laid the foundation of immunochemistry by Karl Landsteiner, an Austrian American biologist, 1868 to 1943, and we basically know him for his discovery of the blood group. So Landsteiner discovered the cause of agglutination to be an immunological reaction that occurs when antibodies are produced by the host against donated blood cells. So the agglutination reaction was basically first propounded by the scientist Karl Landsteiner, and the immune response, and he saw or he observed that there was an immunological reaction when antibodies are produced by the host against the blood donated to him or her. Okay, so this immune response is elicited because blood from different individuals may vary with respect to certain antigens located on the surface of red blood cells. So the concept of the before donation of the blood, the concept of developing to identify the blood group. Why? Because if a person with certain blood group is donated with the blood of other blood group, so there is an agglutination reaction because the body or the anti-red, it recognizes that blood as an anti-RBC blood, and finally it does not recognize it as self, and therefore it finally leads to agglutination reaction in the body. And Landsteiner, with respect to located on the surface of red blood cells, the immune response is elicited because blood from different individuals may vary with respect to certain antigens located on the surface of red blood cells. Landsteiner identified three such antigens, which he labeled as A, B, and C, which was later changed to O, a fourth blood type later named AB. So he basically named it as A, B, and C, which was later changed to O. And again, a fourth blood type which was also later named as AB, was identified the following year. He found that if a person with one blood type, A for example, receives blood from individual of a different blood type, such as B, the host immune system will not recognize the B antigens on the donor blood cells, and thus will consider them to be foreign and dangerous, as it would regard an infectious microbe. Similar reaction which take place by the red blood cells similar to the against the bacteria, and he received the Nobel Prize in 1930 for determining the ABO system of blood groups. Next important work which was done by Gerald M. Edelman 1929 to 2014 and Rodney R. Porter 1917 to 1985, who got Nobel Prize in 1970 2 for their work, and their work was founding the structure of antibody. We know that DNA structure was given by Watson and helix, a double helical structure. Similarly, the antibody, which the students who are familiar with the antibodies structure know that they are basically composed of two chains, heavy chain and light chains. So this structure, this Y-shaped structure, was basically founded by Edelman and Potter, and they were, for their findings of structure of antibodies, they were awarded for their contribution for Nobel Prize in the year 1972. And they found that since our immune system protects us from attacks by microorganisms and poisons, in part through formation of antibodies in the blood that neutralize foreign substances, Gerald Edelman and Rodney Potter separately researched the chemical composition of antibodies using various chemical methods. They split up antibodies into smaller parts and mapped their biological effects. In 1959, this allowed them to determine the structure of antibodies, and antibodies are made up of two smaller and two larger polypeptide chains that form a Y-shaped structure. So these are some of the contribution of some of the important scientists in the field of immunology, and their work gave a very landmark in this field and greatly helped to treat some of the various infections, and also laid the foundation of our modern vaccination technology. I hope you understood this lecture and have gained some important viewpoints on this field. Thank you.